Hello everyone, welcome to the SWAYAM course of Genetics of Fundamental Engineering. Myself Shilpi Banerjee will be today discussing about gene knockout technology. Well, did you know that scientists have the power to turn off specific genes in organisms? Did you know that modification in a single gene can have a significant effect on governing traits or phenotypes? Well, the answer is gene knockout technology. Gene knockout is a technique that selectively turns off a specific gene, and it is basically revolutionizing our understanding of genetics and human health. A knockout, as related to genomics, refers to the use of genetic engineering to inactivate or remove one or more specific genes from an organism. Scientists create knockout organisms in order to study the impact of removing a gene from an organism, which often allows them to then learn something about that gene's function. When I think of knockout, I think of being able to knock out an assignment on my list of tasks, that is to completely finish it and removing it from my list. That is not unlike what we mean when we refer to a gene knockout, where the goal is to make a gene completely non-functional. The simplest way to create a complete gene knockout is to remove the entire region of the gene by cutting it out and then seeing how that affects the organism after removal. This is where the term gene knockout or something like a knockout comes in. This is, however, not the only way to create one, but it is the most complete method. Other ways include changing regions of the gene that are suspected to be important for function. This second method is not always guaranteed to completely interrupt the function of a gene. As mentioned earlier, the term gene knockout refers to the use of genetic engineering techniques which deactivates or deletes one or more of an organism's specific genes. It is also possible to eliminate 2 genes in an organism, or even 3, 4 and more. Such a process is called double knockout, triple knockout or quadruple knockout. Gene knockout involves intentionally deactivating a particular gene in an organism in order to observe the effects and uncover its function. This powerful technique helps researchers understand the role which genes play in development, diseases and many other biological processes. Now, let us look at knockout mice. Laboratory mice are the subject of research into knockout technology. Both humans and mice basically share many common genes, which makes them a suitable species for experiments of such kind. When the test is performed, we can observe a similar result as would occur in a human. Observing knockout mice allows us to understand how a similar gene can cause or contribute to the disease in humans. Knockout mice have contributed to research into diabetes, many cancers, heart disease, arthritis, substance abuse, anxiety, aging and Parkinson's disease. Knockout technology is mainly used to understand the role of a specific gene or region of DNA by comparing a knockout individual to a free living type with a similar genetic background. Knockout organisms are also used as screening tools in drug development to target specific biological processes or to understand the mechanism of an action of a drug. Genes are very much important for us as they encode proteins. These proteins later on participate in different cellular activities and metabolic pathways. Studying a gene's function basically helps us to understand its role in cellular pathways and how it causes abnormalities. Knocking out allows scientists to understand the importance of a gene by functional analysis. Resultantly, they can understand cellular activities, pathways, metabolic activities and various causes of a disease. The first successful gene knockout experiment was conducted by Capecchi et al. In 1980. Basically what they did was they disabled a gene from a mice embryonic stem cells by the process of homologous recombination technique, and the first knockout mouse was born. Capecchi et al. Were awarded the Nobel Prize for Physiology and Medicine for their findings. Now let us take an example to understand this concept in a better fashion. MCH gene encodes mouse coat hair. It's just a, basically this is just a hypothetical example. So mice with the MCH gene have shiny long coat hair. Suppose we wish to understand the importance of the MCH gene in skin cancer. We can use the gene knockout technique to inactivate the MCH gene and understand its consequences in the progenies. We can introduce a mutation in the coding region, remove one exon or inactivate the promoter region. By any means, we have to inactivate the MCH gene and disallow it to encode the coat hair protein. Using a vector, the altered gene can be introduced in the embryonic cell line. This newly born mice which lacks the MCH gene lacks hair on their coat. However, technically it is known as naked mice, but we have explained it to understand the concept of knockout. That's how knockout is performed. Remember, the goal here is to suppress or inactivate a gene's activity selectively. The mice lacking the MCH gene can be categorized into genetically altered or genetically modified organisms, which are known as knockout mice. Now, scientists can observe any phenotypic change that occurs by inactivation. Similarly, the inactivation of two genes is known as double knockout, which was previously explained to you guys. Validation assays are performed to prove gene activation. This technique is popularly used in microorganism and plant genetic research to conduct functional analysis for economically important traits. Now let us look at the process of gene knockout technology. A typical process of gene knockout involves steps like gene selection, vector design and construction, transformation, homologous recombination, selection, identification and validation, insert validation, animal model generation and phenotypic assays. We can basically consider gene knockout as one of the genetic engineering experiment. Now let us look at the details of the different process involved in knockout technology. As mentioned in the previously, first step is gene selection. The very first step in KO is selecting a gene we want to knock out. A gene is selected only if its function is well known and should have been involved in biological activity or biological process we wish to study. Some dry lab work has been carried out to understand the structure of a gene. It is mapped on a chromosome and other parameters such as gene length, number of exons and introns, promoter length and sequence etc. are closely examined in this step. Moving to the second step, that is vector design and its construction. We all know what is vector, right? A vector is nothing but a vehicle that carries our genetic material and transfers it to another location. Viral vectors, plasmids, cosmids and bacterial artificial chromosomes are some of the common types of vectors which are used in gene therapy experiments. Among all these, a plasmid is the most commonly used vector in knockout technology. Plasmid is nothing but bacterial extra chromosomal and circular DNA that can carry the target mutation or sequence for knockout. Next step is which has a vector consisting of the selected marker, a gene specific homologous region and a disrupting element, which can either be a mutation or the sequence. After the construction of vector and validation, our plasmid vector is now ready for transformation. A selected marker or marker gene is used as validation sequence, which is only detectable if transformation or knockout actually occurs. The NEO-R gene, that is neomycin resistance gene, is the standard marker which is used for knockout studies. It is ideally inserted between the left and right arms or between the target sequence, which when recombined with the target genomic region inserts the marker at its location. Such cells can easily grow in the presence of neomycin containing media. Now, let us look at the transformation method. Now, as our plasmid is ready to be inserted into a target cell, generally scientists have been using selective embryonic stem cells for transformation. Basically, embryonic stem cells are more commonly used because they can divide at a much faster rate into different cell types and form various tissues. The vector once finds the gene specific target location, it immediately recombines and inserts our target change with the marker gene in the target cells. Transformed cells are then cultured using an appropriate media. Electroporation, sonication and microinjection are some of the commonly used techniques for ESC's transformation. Note that cells will only grow if transformation occurs properly. Now, let us look at homologous recombination. After successful transformation and after reaching the complementary region, homologous recombination will occur, which results in the disruption of a gene's function later on. Homologous recombination introduces the change that eventually knocks out that particular targeted gene. Moving on to the selection and validation. It is very important to validate whether the transformation has fully taken place or not, which means the experiment has been successfully placed or not. Techniques like polymerase chain reaction and DNA sequence can validate the process of knockout technology. A few transformed cells are taken for DNA isolation. DNA sequencing can validate KO at the sequence level. PCR and qPCR can also be used for validation using sequence specific primers. qPCR helps to determine the concentration of the target gene from the transformed cells, and it also determines if knockout has occurred or not. Besides this, restriction digestion can also be employed for KO verification, but this is much less recommended. After the completion of validation process, the successfully transformed cells are now selected for the generation of genetically modified model organisms. Now, let us look at the introduction into the animal model. Now, the transformed cells are employed to generate a KO animal using breeding and other techniques. Phenotypic investigation involves physical, biochemical and functional investigations. In vitro experiments are carried out to study the effect of gene loss. Various chemical assays are also available for protein studies as well. As you can see in this pictorial representation, that how knockout technology is performed, which has been explained in the previous steps. Moving on further, let us look at gene knockout methods. So far we have discussed the KO experiment in context with the homologous recombination. However, there are other methods as well that scientists can use. Gene silencing, conditional knockout, gene editing and knockout by mutation are common methods. We will discuss each technique one after other in this section. But remember, in any of the method, the purpose is to inactivate or make silence the target gene via, that is, knockout. Gene silencing. Gene silencing basically aims to deactivate a gene by disrupting its mRNA. A cell's natural gene regulation mechanism uses RNA interference. Here, small complementary microRNAs bind with the mRNA and deactivate them for translation. During in vitro gene silencing, scientists use artificially synthesized small interfering RNA complementary to the mRNA of the target gene. Upon reaching the target location, it binds with the mRNA, a process in which RICSC complex, much like the cell's natural mechanism, and disallows translation. This process blocks protein translation from the mRNA by mRNA disruption and effectively silences the gene. That is, basically the gene is knocked out. Other techniques like antisense RNA are also used. Now let us look at what is conditional knockout. Conditional knockout is specific. It allows the inactivation of a target gene in a specific tissue or specific developmental stage, or gene activation has been carried out to study a specific function in a specific cell population, tissue or developmental stage. Conditional knockout is a great tool to study tissue specific cancer. The present technique uses homologous recombination and tissue specific promoter for conditional knockout, where knockout only occurs in selected tissues or cell. CKO is the best way to study a gene's function at a tissue or a cellular level. Interestingly, it is not only performed in the embryonic stage but also on adult animals as well. Cre-LoxP is one such technique in which the CRE combines recombinase works on LoxP tissue specific inactivation. Recent studies have showed that CKO is widely used in breast cancer and ovarian cancer research. Conditional knockout is very popular and by far the most successful technique for gene inactivation. The Cre-Lox system is one of the most powerful and versatile tools developed for mouse genetics. It gives mouse researchers sophisticated control over the location and timing of gene expression. Cre-Lox is usually used to make knockout alleles, but it can also be used to activate gene expression. Now let us look at gene knockout by mutation. Knockout by mutation is yet another important technique. In this technique, a mutation is incorporated in a target gene or sequence in a way that deactivates or alters the target gene's function. Such mutation is known as loss of function, which can be either insertion, deletion, duplication or translocation. Artificial mutagenesis techniques like chemical alteration, radiation based mutagenesis or physical mutagenesis have been used to incorporate a mutation. Now let us look at gene knockout by gene therapy. Knockout by gene therapy is a novel approach which involves the delivery of a therapeutic agent, engineered vector or system to suppress a gene's function. CRISPR-Cas9 is a popular and modern gene therapy technique that can introduce a mutation or remove a sequence from the target gene. Any target element, for example, the sgRNA in case of CRISPR technique is engineered in such a way that introduces a desired change in the DNA sequence. Such techniques are very much accurate and precise for gene knockout. Gene therapy approaches holds promise for potential applications in treating genetic diseases. Now, let us look at homologous recombination. Homologous recombination involves the exchange of genetic material between two homologous sequence or genes. Homologous recombination has a higher success rate. Thus, scientists use this phenomena for KO. Here, scientists prepare homologous sequences complementary to the flanking region of the target gene and introduce them in the vector. The vector then transfers it to the target location, where the recombinase enzyme performs recombination and exchanges the native sequence with a target sequence. Interestingly, this process either disrupts a gene function, eliminates it, or suppresses it completely. Make a note that, as mentioned earlier, a marker gene is also incorporated for validation. This pictorial representation basically shows how a vector is used for, how a plasmid vector is basically used for knockout technology. Now, let us look at some of the applications of gene knockout. The key function of knockout is to eliminate or inactivate a gene, thus making it functionally inactive. The present revolutionary approach has opened many doors for researchers to study diseases, genes and traits. Here are, let us basically look at some of the amazing and important applications of gene knockout. The first one is to understand a gene's function. As we have discussed so far, that the pivotal function of KO is to understand a gene's function. Scientists can study alteration in phenotypes or traits by selective knockout or target gene inactivation. KO experiments on model organisms help to study genes' role in development, behavior, biological activities, physiology and disease progression. Eventually, such studies contribute to develop new strategies and therapies for diseases. Next application is the relation, to understand the relation between genotype and phenotype of an organism. Gene knockout is a great tool to establish the relationship between a gene and a phenotype. Through selective gene inactivation, its effect on a phenotype or group of phenotypes can be studied. This way, scientists understand the relationship in much better way between a gene and a phenotype. Now, let us look to the application of knockout in functional genomics. This is functional genomics is largely defined as to understand the overall function of the genome. KO is a crucial tool in functional genomic studies. Through selective gene targeting, scientists can systematically investigate the impact of individual genes on related functions, pathways, cellular activities, signaling pathways and many others. Scientists can understand the function as well as the evolutionary importance of homologous genes and genes families. Now, let us see application of KO in evolutionary studies. This is gene knockout is an amazing tool for studying evolution and the role of a gene in evolutionary events. By selectively inactivating a gene across different organisms, scientists can understand a gene's effect and its evolutionary importance. This helps to understand the underlying mechanism of evolution and biodiversity for studies of various diseases. KO also helps to understand the molecular mechanism behind diseases through gene level analysis. Scientists prepare animal models to perform KO for a particular disease governing gene and understand the effect. These studies help us understand how genetic diseases happen and how certain genes or groups of genes play a role in causing disease. This in turn helps to understand creating new treatments for a disease. Next is Drug studies and development. Assessing a disease at the molecular level and using gene knockout techniques also helps to understand the response of a drug. This means that scientists can evaluate the effectiveness, efficiency and response of a drug when targeting specific genes or when those genes which are not targeted. Such valuable information provides value in drug development, studies and discovery. It also helps to strengthen the therapeutic approach against a particular disease. The next application is developing gene therapy. Gene knockout recently has given more strength to the field of gene therapy. New therapeutic approaches and gene therapy treatments can be developed against particular or many gene defects. Various CRISPR and Cas9 therapies mediated by KO are now under clinical trials and are ready to rule the world. Other therapies are also under research. Agriculture and livestock improvement. Not only in disease studies and human genetics, but gene knockout technique also have great value in the agriculture industry and livestock improvement. Through knockout studies, researchers can understand the function of an economically important trait and thereby can enhance it. Desirable and economically important traits like disease resistant, yield improvement, nutrient values in plants and growth, development, milking capacity in animals can be improved. Now, let us see about the limitations of gene knockout. Well, we all know that every coin has two sides. Though gene knockout is highly useful and precise tool in the field of genetic engineering, it also has certain important limitations. Now, let us look at some limitations of gene knockout. Off targeting effects. Any gene knockout experiment is designed to specifically inactivate a target gene. However, many times it can suppress or affect other genes' activity unintentionally, or in many cases it inactivates entirely a different gene. These two scenarios are known as an off-targeting effect. Research shows substantial off-targeting gene knockout effects which may directly or indirectly impact the function of other genes or regulatory elements. Gene compensatory effects. Compensatory mechanisms may exist for several genes that compensate or fill the function of inactivated gene. This particularly occurs during the developmental process in which KO triggers the developmental compensatory mechanism. Resultantly, even after successful gene knockout, the function will remain intact. Such events pose challenges to understand the full potential effect of a gene and its governing traits. Now, let us look at the limitation, which is basically a compensatory action. In some cases, redundant genes can compensate for the effect of a target gene. Therefore, simply knocking out a single gene may not result in a loss of function. Other genes known as redundant genes can step in and continue to produce the expected traits. These compensatory effects of genes can restrict the effectiveness of gene knockout. Gene knockout for essential genes. There are a set of genes in organisms that are essential for their survival. Alteration in such genes can cause lethal effects or genetic abnormalities. Scientists cannot manipulate every time and study these genes by gene knockout. Tissue specificity. Gene knockout has less tissue specificity as it can impact a broad range of cells and tissues. So, while targeting a specific gene for specific tissues, adequate results cannot be obtained always. Now, let us see at the concept of gene knock-in as the reverse of gene knockout. Interesting, right? So, in genetic engineering, this process involves the insertion or replacement of one gene with another which is not normally present in the target organism. Through this process, we can obtain a model organism to study aspects of a specific disease. Gene knockout is an important technique in genetic engineering that helps scientists learn about the function of specific gene and how they work. It allows us to understand the mechanisms behind gene function. Although there are some limitations like unintended effects, lack of specificity in tissues, compensatory effects, scientists are actively working to overcome them. Gene knockout is used in various fields such as agriculture, disease research, drug development and other studies. Now, let us look at some of the future prospects and development related to gene knockout. Advancement in gene editing technology. The field of gene editing has seen many remarkable advancement with techniques like CRISPR-Cas9, which takes the center stage. As these technologies become more refined and accessible, the precision and efficiency of gene knockout will improve. This will enable researchers to target specific genes with greater accuracy, reducing their off-target effects. Moving on to the next, as mentioned earlier, the concept of functional genomics. Gene knockout plays a crucial role in functional genomics, allowing scientists to understand the role of specific genes in disease, development and normal physiological processes. Future research will likely involve large-scale knockout studies to uncover the functions of the thousands of uncharacterized genes in the human genome. Next is therapeutic applications. Gene knockout has the potential to revolutionize medicine. It can be used to treat genetic disorders by disabling or correcting malfunctioning genes. For example, ongoing research is going on which explores the use of gene knockout in treating conditions like cystic fibrosis and sickle cell disease. Next is cancer therapy. Gene knockout can be used to target and deactivate genes that drive cancer growth. In the future, personalized cancer therapies could involve the knockout of specific oncogenes in individual patients, leading to more effective and less toxic treatments. In agriculture, gene knockout can be used to develop crops with desirable traits such as disease resistance, improved nutritional content or reduced environmental impact. This technology may also be employed to combat plant pathogens by disabling their virulence genes. Next future advancement is bioproduction. Gene knockout can enhance the production of biofuels, pharmaceutical and industrial enzymes. By knocking out genes that interfere with production pathways, researchers can engineer microorganisms and cell lines for more efficient and cost effective production processes. Next is human enhancement and ethical concern. As gene editing technologies advance, ethical consideration become increasingly important. Future debates will focus on the potential misuse of gene knockout for purposes such as human enhancement, designer babies or creating genetically modified organisms with unforeseen ecological consequences. Gene therapy for neurological disorder is also one of the most promising aspects of knockout. This can be used in the development of gene therapy for neurological disease like Huntington, Alzheimer. By silencing or correcting these causing genes, it may be possible to slow or even halt the progression of the disease. This knockout can also be used as a regenerative medicine. In regenerative medicine, gene knockout can be used to enhance the safety and effectiveness of stem cell therapies. Researchers are exploring new ways to remove or deactivate genes associated with tumor formation or immune rejection to improve the success of transplantations. For environmental conservation, gene knockout could be used to control invasive species or mitigate the environmental impact of certain organisms. However, this raises significant ethical and ecological concerns that will need to be carefully considered. While the prospects for gene knockout are promising, it is essential to approach these advancements with ethical consideration, responsible research practices and careful oversight to ensure that the technology is used for the benefit of humanity and not the environment. In summary, Gene knockout, as you can see, is a powerful technique in molecular biology and genetic engineering, which involves the deactivation or deletion of a specific gene within an organism's genome. This technique has had and will continue to have significant implication for scientific research, biotechnology and many other applications. With this, thank you everyone. I hope you could understand what is knockout, their steps, their application and its future prospect.